The caption here says, "And notice, gentlemen, the faster I go, the more Simmons sounds like a motorboat." Not entirely sure what aspect of science that underwrites, but there you have it. A lot of Far Side cartoons. Controls are not really controls. And a controlled experiment isn't a control that you really got your hands around. It has a different, it's the way it works. We take English words and we use them for something specific that it makes it confusing. A control is your comparison. It could be one test where you've changed the variable in a known way and then you look for changes in your results that you can connect to the changes in the variables. A classic control is a test where you can take the variable away completely. We do that with drug tests, where you give one group the drug and you give another group a fake version of the drug where the only thing really different is that chemical that you're testing. But the assumption is that any difference in results between the two tests is connected specifically to that chemical and not to the way you're treating them or anything else. But a lot of times you can't do that. You can't completely remove a variable because it isn't removable. You can't run, say, a test on the effects of temperature and have your variable be temperature where you have a test where there's no temperature. You can change the temperature and then look for results that associate with temperature going up, temperature going down, that sort of thing. And if you're testing plants and you're using different issues of light, you can run a test with no lights, but it's not really a practical. They're gonna like some light. So probably running a control with no light, somebody might go, you needed to do that. No, we were pretty sure we knew what was gonna happen if we raised some of our plants in a closet. The biggest issue you wind up with when you're designing your experiment is, so what's going on that's gonna mess with your results, gonna change them significantly, that you're not actually testing. Those are confounding factors. Confounding factors are going to influence what's going on in your test and what you get at the end, but they're not actually the variable. And you always have to be on the lookout for that because sometimes you don't realize you have confounding factors until you're halfway through the experiment. And then you have to decide whether you can factor them in somehow or go back and start all over again with some sort of redesign that takes into account confounding factors. And sometimes you wind up doing side tests where somebody goes, well, isn't this gonna mess with your results? And you go, wow, I don't really know. Let's run some tests over here and change those factors and see if they do have an effect on the results. So you might run parallel tests just to see if a confounding factor is actually something you needed to take into account and didn't, or it actually isn't doing anything. A famous confounding factor is placebo effect and all the variants that you can connect to this that aren't really placebo effect. The original version of this was people running animal tests. So you're testing a drug. You've got a whole bunch of mice that have the condition that you're interested in. And you're going to give them an injection to try to make them better. So you've got your sick mice over here as controls and your experimental mice that you're injecting every day. And somebody in the group went, does it matter that we're not injecting the control mice? we don't really know how taking and handling them and giving them an injection is affecting them in a way that may be changing our results. And somebody goes, yeah, you're probably right. And we're gonna have to do the same thing to the control mice that we're doing to the experimental mice. But we can't give them the chemical, we gotta give them a substitute that we already know should not do anything to them. So just a saline solution that matches their tissue saline. But yeah, we're gonna grab these guys every day and give them injection the same way that we're giving the experimental group an injection. And that idea of the treatment itself producing effects, it also came up when people started to experiment on humans. And it's like, what effect does actually being treated have on the average patient? Is it inclined to make them feel better in some sort of significant way that's going to change our results? And the assumption is that it depends on what you're testing. By better, does it mean, is it going to cure their cancer? No, probably not. But if you're running cancer tests, part of what you're testing is quality of life. And that is how they feel. And the placebo effect may definitely change that if you're treating your experimental group and you're just letting your control group just die of cancer, but you're testing them on their quality of life. The fact that the cancer people are being treated may make them feel better. They don't feel as sick, they don't feel as nauseated, is that they don't have, either they're not having the same headaches or they're not noticing the same headaches. You need to give your control group the same treatment, and nobody in the groups should know whether they're getting treated or not. That's blinding. And there's 3 levels of this. We're administering this to the group, but the people in each group don't know what they're getting. They're getting a fake, the control people are getting a fake treatment that matches the real treatment, except it doesn't have the chemical in it, which of course means sometimes they can figure out which group they're in. There's a confounding factor. But you can come up with ways around, but then you run into another confounding factor of ethics. Artificial side effects in a placebo is an iffy thing. Some people have done it, but it's an iffy thing. The patients not knowing which group they're in is blinding. They did that for a long time, and in the 1960s, someone suggested that the administrators of the test, the doctors who were actually treating the patients, might be treating them differently if they knew which group they were in. And they did, and then it was a matter of, well, how do you test that? What do you measure? And they decided that, okay, if I know that you're getting a drug, what am I expecting from you? I'm expecting you to get better, I'm expecting side effects. If you're getting a fake version of the drug, I'm not expecting you to get better, I'm expecting you to get worse. Are there certain ways that we're talking to our patients that would indicate and subconsciously let them know what we're expecting? And so they had people track doctors in other studies who didn't know what was going on, looking for discussions of side effects, discussions of worsening symptoms, discussions of those items that they could actually count. What do you measure? And they found out that, yeah, the doctors were treating the 2 groups somewhat differently, is that the much more talk of side effects in the test group than in the placebo group. Much more talk of worsening symptoms in the placebo group than in the test group. So they decided we can't do it this way anymore. And now we have double-blinded tests where not only do the patients know not what group they're in, the people treating them don't know which group they're in. The company assigns everybody a number and then randomly assigns experiment control to the numbers. And then you get the treatments with your patient 108, you're getting this treatment 108. I don't know if it's the real treatment or not. So the doctors can no longer influence the patients, and the fact that the drug company knows they're not having contact with the patient, so they can't produce a placebo effect. I said there are 3 levels. A few years ago, someone started to wonder, these tests are getting very, very complicated, broken down by age and ethnicity and a whole bunch of comorbidities and stuff. And so there's a lot of number crunching and comparisons that go on. And someone went, So our numbers people, there are things that you do with the numbers that are somewhat subjective. Does it matter if they know whether they're doing the numbers on the experimental group or the placebo group? And somebody went, I don't know. But we can blind that. So triple blinded studies, the number crunchers don't know which group they're manipulating, they're tracking the data on. So if there is a way for them to kind of skew, you know, the experimental group to look like it's more effect, or the placebo group to look like it's less effect, they don't know to do that because they don't know which group that they're doing. So that's blindedness in relation to the placebo effect. A placebo effect is an example of an artifact. These can come up in all sorts of different designs. An artifact is a result not from your variable, but from the way that the test itself is run. When you're treating people is part of the test design. It's gotta be, and it produces a placebo effect. Because of that, it's an artifact. Another example I give is that 40 years ago, something like that, when people were first starting to try to figure out how Alzheimer's works, and one of the questions was, are people getting some sort of chemical that's depositing in their brain from the environment that's causing Alzheimer's? So one of the researchers was taking and testing slices of brain tissue from Alzheimer's patients who had died, and testing slices from controls who died at similar ages and had as many other confounding factors like general health and other kinds of things that kind of matched the Alzheimer's patients. And he looked for different environmental chemicals in the tissue samples. And he found one of the environmental chemicals that was in the Alzheimer's brains that was not in the control brains was aluminum. And so he published a paper on that. People threw out their aluminum pots and they started to rethink how aluminum cans get made. They're sealed on the inside, so aluminum in cans don't tend to bleed aluminum into the beverages. But it became a really, really big deal. Now what also happens in science is people take your results and use them as a starting place for more study. So somebody went, well, let's see what the aluminum is doing to the cells. And he couldn't find the aluminum. And he couldn't find the aluminum, And he finally, a little clue, he sent a note to the original researcher saying, when you publish your results, you don't put every single detail of your study down. There's just not room for it. And so he said to the guy, could you tell me more about how you prepared your tissue samples? Because I'm doing my tissue samples and I'm not finding any aluminum at all. And the guy gave him the list, and the second guy goes, oh, you're using a different preservative for the tissue than I am. That preservative has aluminum in it. I just ran some of my tissue samples through that preservative. I'm getting aluminum. It's not there to begin with. You're actually putting it there. Is that the difference is that Alzheimer's brains, when you expose it to the aluminum preservative, it hangs on to it. Normal brains, when you expose it to the aluminum containing preservative, and then you rinse them, the aluminum rinses away. So the only reason there was aluminum in the Alzheimer's brains literally was because the researcher put it there. It was an artifact of the way he was doing his test. And so any kind of result that you can connect to your process, hopefully you can change the process to make that go away and not have it be a confounding factor to your test. What's in the news now is a lot of studies for things like hydroxychloroquine or other kinds of things that we think might have an effect on the development of disease in coronavirus patients. And they'll go, oh yeah, it works. And the people that know how this works, the peers look and go, it's, number one, it's not a blinded double group experiment. And also, they only had like 12 people they were looking at, or maybe 30 people. And the thing is that when you have a small group of subjects, then subjects that have unusual responses will swing your average much more significantly than if that same subject was in a group of 1,000. They're in a group of 8, they're swinging that average off toward their extreme, whatever it is. They're in a group of 1,000, they're not moving your average off of the real average very much. So you have to try to design around the impact of chance. You don't want to study too few subjects or run a test too few times because you can't trust those results. And that's factored into what we call significant data, significant differences. So the best way to get around this is high numbers. You can't always get high numbers in the real world. Sometimes if you're running, say, a drug study on a fairly rare disease, is that you may not be able to get that many patients that do it. You may get so few patients that you can't really, it makes no sense to split off a control group because you can't really trust the numbers anyway. So a lot of what they call orphan disease tests are done in a not classic scientific method way. But you want to run lots and lots of numbers because what you're trying to avoid is something called anecdotal evidence. So you go, oh look, the hydroxychloroquine works. And somebody goes, well, that's just anecdotal. Now in English, that means it's connected to a story of yours. And you know, when you go, you know, my great uncle Harry, you know, chewed on pine bark and his cancer went away, that's anecdotal. That you know, maybe there's something there, but it's not a lot of evidence that pine bark was effective. You also don't know a lot more about Uncle Harry. But if somebody does hydroxychloroquine and they only had 8 subjects, somebody can very easily go, well, that's anecdotal. Is that the numbers aren't trustworthy, they're too small. And in classic science, anecdotal evidence is just too limited. It's not that it's not evidence, it could be evidence, it could mean something, but you don't really have the really good evidence. If people they may go, oh, that's not proof. That's true, but we should be avoiding that term. Guys at a blackboard, you guys, I tend to agree with you, especially since 6 times 10 to the, and then there's a whole bunch of stuff, is my lucky number. A huge confounding factor is bias. Is that you're testing your own idea. It's really easy to see evidence as positive because you're expecting it to be positive. It's easy to ignore stuff that maybe is pushing the other way or decide that it's worth ignoring. You go, oh no, that stuff that happened over there, here's the reason why I didn't count those. Because I didn't really think that they were valid, and you come up with ways. You try not to. Is everybody should be trying to avoid their own biases when they're designing and running and gathering data for their experiments. There's a term that connects to this that has like 16 different definitions. But here, it's the idea that things that are done by people involve the influences of what the person believes, how they were raised, the culture that they're in, is that what's already known, what they were taught as kids. When Darwin left England and traveled the world and saw people of different ethnic groups, is that he was, at least as I read it, he was trained that brown and black people were actually not Homo sapiens. They were different, not just different races, but different species. And people did use that as a justification for slavery, like, they're not really people. But as he traveled the world and came in contact with these cultures, he came back going, no, this is totally wrong. But he didn't know any better. That was his bias to start with. And you'll see old scientists that say these horribly, what seems like horribly racist things, And a lot of times, it's just pure ignorance based on what they had been taught going in. They said they didn't have the experience to think otherwise. And that's a really extreme example, but bias can work a lot of different ways. It's like the nurses at the, in the neonate units, is they were absolutely convinced that it was crazy to test this thing about crack babies, because crack babies were definitely different than regular babies. They saw it all the time. They were seeing what they expected to see. And postmodernism is that influence. And postmodernism works in, you'll hear this expression, the winners write the history books. Our history books would have a very different skew on Nazi Germany if they had won the war, compared to how that we skew that with we winning the war. So history runs through postmodernism. This was originally a philosophical and an artistic idea, where art started to look in terms of integrating the artist into the art. So there's, like I said, there's a whole bunch of different applications for postmodernism, but here it's part of how much of you is working its way into your design in a way that's not necessarily good. Connected to this is the idea of ethics. Well, you could do that, but you really shouldn't. People do, 2 years ago, someone doing a test of gene editing did it on a couple of human embryos. Really isn't at a level that you should be doing that, and everybody thought that it was wrong to do, and he's gotten in a lot of trouble. He did talk to some people, he talked to some peers about it, and they went, no, you really shouldn't be doing that. And they're in a little bit of trouble because they told him not to do it. They were very firm in that. And he went off and did it. They didn't really follow up, and they've gotten in a little trouble because they didn't report it, or they didn't, get more forceful about it. But it's ethics, is that there are rights and wrongs that are partly postmodernistic, influences of your culture and your society. And, don't be doing that on endangered species sort of things. You shouldn't be doing these kinds of tests on humans. You shouldn't be doing these sorts of tests on animals. There's a lot of ethics in animal testing of what you should or you should not do, or what level of benefit against the cost of what you're doing to the animals is. And that has a huge influence on design. Okay, we're going to stop there, pick it up from here.